tumor site. And of course, there's a problem with the, the tumor immune suppressive microenvironment. And when the immune cells, the effector cells get there, and they are suppressed in terms of their effector functions. Of course, there toxicity associated with T cell therapy, like cytokine stops. Okay, so why we want to think about using NK cells? They are killer cells. They're called natural killer cells, and they can have direct cytotoxicity against tumors. And their cytotoxicity does not involve specific antigen recognition in the context of HLA molecules. They can either directly recognize the target molecule, sometimes the stress-induced molecules expressed by tumor cells using NK activating receptors, or they can kill the target through, we call it ADCC, antibody-dependent cytotoxicity, through the Fc receptors like CD16. So during NK maturation, they actually becoming more and more cytotoxic. So in both human and mice, when they mature and get to the periphery, you can see they lose proliferative capacity and gain their killing activities. So yeah, I also want to mention that they have high CD16 expression, which is the Fc receptor, and they also have a lot of cytotoxic granules in there, so they become better killers. So in terms of NK function, I want to briefly introduce to those of you who are not, really into immunology, other than their direct killing effects. Okay, they can also have immune regulatory functions. They can be activated by dendritic cells through type 1 interferon or IL-12. And in turn, they can also work on dendritic cells by activating them to go through their maturation, and also they can promote DC migration. And those dendritic cells being activated by the NK cells can also improve their cross-priming activity to activate CD8 T cells, and the interferon gamma produced by the NK cells can also promote Th1 polarization in the CD4 T cells, which is very beneficial in the tumor settings. So a lot of NK adoptive therapy have already been done in the hematological stem cell transplantation settings. We have gained quite a lot of experiences there by using donor alloreactive NK cells. So those alloreactive NK cells can either directly lyse the host leukemia cells here, or they can kill the host dendritic cells and the host T cells in order to promote engraftment. And they can also regulate donor T cells in reducing graft versus host disease. We had a paper published a couple of years ago looking at cytokine pre-activated NK cells, where we found the IL-12-18 and also the IL-12-18-15 combination can both promote graft versus leukemia effect during transplantation, and they can also inhibit severe GVHD, acute GVHD. But in terms of mild acute GVHD, we can see that the 12-18 activation can inhibit or reduce mild AGHD, but the triple activated NK cells can actually promote GVHD. So in this setting, it's a bit interesting to further look at the NK cell functions in the transplant setting, because a bit differences here can make actually a big difference in the outcome, and could be opposite effect. So, and in the tumors, it may not be that complicated, and we don't have to think about the GVHD problem because we're using, autologous NK cells. So there are also publications suggesting, although NK cells are considered to be innate cells, there is NK cell memory. So in both human and mice, people have found in certain viral infections, there are some viral antigens that can specifically expand certain subsets of NK cells which have memory functions, because the response can be significantly heightened during the secondary infection. Also in the cytokine stimulation settings, both human and mice, and there are memory-like NK cells. They are not antigen specific, but if you stimulate them with this cytokine combination with IL-12, 15, and 18, and then you can use IL-15 alone to re-stimulate, and you get a heightened NK memory response. So it's called memory-like. So this memory-like NK cells have been used actually in the adoptive therapy setting by adoptively transfer those cytokine pre-activated NK cells or memory-like NK cells. And they have significantly higher interferon gamma production when they are co-cultured with AML cells, either primary AML or K562 cell lines. And if you use those cells in the NSG mice model, and you can see those memory-like NK cells can significantly inhibit tumor or leukemia development and prolong the survival of the mice. So this is an area where NK cells can be manipulated to be enhanced in terms of their anti-tumor functions. So what are the consensus about NK cells in solid tumors? So if you look at the the number or the percentage of the NK cells in the solid tumors and correlate that with either 5-year survival, overall survival, or disease-free survival, and then you can see either people found no impact or they found positive impact there. So in general, there's a positive correlation between the presence of NK cells and the overall outcome of the cancer patient. So which is a good news. And then people started early on, actually from the 80s, to target NK cells for cancer therapy. In the early days, when people used LAC cells, the lymphokine activated killer cells, they are actually mainly NK cells and NKT cells. People have used those for treating advanced metastatic cancer. And then later on, in the 90s, people started, Prof. Dario Cabana is the one that pioneered in that area using CAR NK cells and had a lot of successes in leukemia, lymphoma. And then in the late 90s, there are ideas of using bispecific killer engagers, the BiKEs, trying to engage NK cells better in cancer therapy. So during the development of the checkpoint blockade in the recent decade, NK cells, have been used in clinical trials derived from PBMC or iPSC, the induced pluripotent stem cells. And also there are NK CAR T cells developed from iPSCs. They're all in clinical trials. So what are the source of the NK cells being used currently in the clinical trials? There are, one of them is NK92 cells. This is a human NK cell line, has been very useful, and people use it for research, and also now, and there are multiple different modifications of the cell lines, and people trying to develop kind of an off-the-shelf NK product for cancer immunotherapy. And as I mentioned, it can be derived from blood and stem cells or cord blood. Okay. So what are the modifications that are being done now? Of course, people are using CAR There are also, other than CAR, there's some other genetically modified NK cells, for example, using high affinity CD16. There are also a group of NK cells that are called adaptive NK cells. They are naturally expanded CMV-specific NK cells. And there are also cytokine-induced memory-like NK cells, like what I mentioned, using interleukin 12, 15, and 18. However, similar to the CAR, the alpha-beta T cell therapy, NK cells face similar situations in the tumor microenvironment. So the cytokines such as TGF-beta can also function on NK cells to reduce NK cell activation. And there are mechanisms to reduce NK activating receptor expression. And there are also molecules in the tumor can induce NK apoptosis. Of course, there's also checkpoint blockade mechanism to induce NK exhaustion. So there's still quite a lot need to be done to enhance NK cell function. This is a busy slide. I hope you don't get panic. I just want to point to a few points here. One is that, similarly, NK cells also express PD-1. So anti-PD-1, PD-L1 antibody is supposed to be also working here. They also express NKG2A, KIA, which are inhibitory receptors of NK cells, which can be used to block, to enhance NK function. There's also, a lot of efforts in enhancing IL-15 signal by making, superagonist IL-15 to stimulate through IL-15 receptor. And there are also BIKE and TRIKE. So this is the tri-specific killer engager linking tumor antigen, here is CD19, with IL-15 receptor together with Fc receptor CD16. So quite a lot of efforts are being made here trying to engage NK cell better. Lastly, monoclonal antibodies can be used to target NK cells to enhance ADCC by linking a tumor antigen with CD16. So the NK cell engagers, as I mentioned, those are the ones that are in clinical trials. For this one targeting CD33, this one targeting CD30. This is a bispecific, it's a BIKE, and also this one targeting HER2. So as I mentioned, for cytokine therapy, mainly targeting IL-15. There's superagonist IL-15, recombinant IL-15, and also a soluble IL-15, IL-15 receptor alpha heterodimer. Okay, so here just want to impress you that in the T cell field, there are a lot of T cell studies focusing on different T cell subsets. Okay, so both CD4 and CD8. So the question is, are the NK cells just a one single killer cell, or there are also different function or different subsets within the NK population? And we know very little about that. So I'm going to show you some research from my lab and trying to understand different populations in the NK big NK family. And I think only when we understand better here, then we can try to design better NK cellular products for cancer therapy. So we did a single cell sequencing, looking at all the NK cells in the tumor microenvironment. So of course you have conventional NK cells, and this is in the liver, and in the liver cancer context in the mouse model. So if you look at in the liver, there are a population called liver resident NK cells. And you can see the conventional NK cells are quite similar, a lot of clusters together here. The closer they are, the similar they are. And then for the liver resident NK cells, we have three clusters. So 1 to 3 are very close together, but they're very far away from this cluster 2. So if you look at the gene expression profiles and you see the similar thing that 1 to 3 are quite similar, but 2 is distinctly different. Okay, so then we asked the question by looking at different pathways. What are the difference in the pathways of those clusters? So the top one is phospholipid binding. So we look at some of the scavenger receptor expressions which are responsible for binding to the lipid, and they are highly expressed actually in the class 1 to 3, but low in cluster 2. So we compare the conventional NK and the liver resident NK population. That you can see the liver resident NK cells have higher lipid contents, and they do have higher expression of the scavenger receptor, the CD68. And also, if you compare tumor and non-tumor, and you do see in the tumor, each line represents one mouse, and they do have higher expression in the tumor compared to the non-tumor liver, where for the conventional NK, there's no difference here. So then we got interested to look at, whether those, liver resident NK cells, which are high in scavenger receptors, are really liver resident cells, or they are derived from other conventional NK cells. So with the help of Veronique Angelis' lab, we did this called parabiosis, where you link the 2 mice together. So if you look at the CD45.2 mice, so all the CD45.2 positive cells are resident in this mice, and the ones that are CD45.2 negative are actually coming from the pair. Okay, so we look at the conventional NK cells, which is CD49B positive. You can see half and half. So half is from the host and half is from the pair, because we link their blood vessel together. But if you look at the resident cells, especially the CD68 positive cells, and almost 100% of them are actually CD45.2 positive, suggesting those cells actually host resident in the liver. They're not coming from circulation. So we also look at the human atlas established by Dr. Chen-Jing Miao from SIGN. And if you look at CD68 expression, there are 2 NK cell populations distinct in their CD68 expression. So then we became interested to look at, what is the cells are doing in the tumor, right? So we dissociate the liver into tumor site, peri-tumor site, and normal liver. So we compare them in terms of the CD68 positive cells. And you do see, in the conventional NK cells do not have any expression, but in the liver resident NK population, that is higher in the tumor. So you do have accumulation of those NK cells in the tumor. So, and then we want to ask, what kind of functions are they when those cells increase in the tumor? They do have higher expression of the scavenger receptors other than CD68. They, LAG3, for those of you who are not familiar with immunology, it's a molecule that's similar as PD-1. It's an inhibitory molecule on the surface of the T cell and the NK cells. So you can see in the tumor, LAG3 is higher, and also the CD68 positive liver resident NK cells have even higher LAG3 expression. So PD-L1 is higher in liver resident NK cells, both CD68 positive and negative. In terms of cytokine production, okay, granzyme B is important for killing. You can see it's decreased in those CD68 NK. The TNF alpha and interferon gamma are supposed to be type 1 cytokines that is good for that are good for anti-tumor immunity. It's reduced in the CD68 positive liver resident NK cells. Interferon gamma is also reduced, and those cells express high IL-10 which is supposed to be, immune inhibitory in most of the cases. So overall, this subset of liver-resident NK cells do have some kind of immune suppressive phenotypes. And they might be the cells that go into the liver and being modulated and suppressed by the tumor microenvironment. So here shows again, compared to the blue and the green. So the green part is the CD68 positive liver-resident NK cells. They have reduced granzyme B and TNF and interferon gamma, but have increased IL-10. So the killing, this is not a very good killing assay because we had difficulty getting enough cells. But it's quite obvious that you have killing capacity from the CD68 negative cells and also the conventional NK cells, but you completely lost the killing in this CD68 positive liver-resident NK cells. So I'm just going to quickly summarize the NK part, what I have told you so far. So NK cells are promising cellular products for cancer adoptive immunotherapy. The tumor immune suppressive microenvironment poses significant challenges to NK-based therapies. And NK cell engager molecules and cytokine-based treatment are the current major strategies enhancing NK cell functions. And better understanding of the NK-based cancer immunotherapy, NK subsets and the regulation of their functions are critical for the future NK-based cancer immunotherapy. So there's still quite a lot to do, because now we can see by single cell sequencing, RNA sequencing, it's not just one single NK population. There are many subsets there. And then the tumor definitely has a big impact on the NK phenotypes and functions. Only if we understand that, and then we can design better therapies for NK product. Now I'm going to switch gears about halfway done to talk about gamma delta T cells. So there are two classes of T cell receptors expressed on the surface of T cells. So you probably know the major one is alpha beta T cell receptor, and then there's also gamma delta T cell receptor. So the gamma delta T cells, are present in a lot of our tissues, and they can be found in peripheral blood, lymph nodes, spleen, etc. And again, I think it's due to our quite minimal understanding of those cells, because a lot of effort have been put into alpha beta T cells. They are the subsets are quite blurred, not as as clear as alpha beta T cells. So for human, mainly we know that there are different V delta populations. Okay, so the V delta 2, V gamma 9 are the major gamma delta T cell subset in the peripheral blood, and then the ones in the tissues are mainly the V delta 1 gamma delta T cells. And for the mouse, it's mainly subdivided with the V gamma chains used. Okay. So similar to NK cells, gamma delta T cells have been considered to be innate cell for quite a while, because they do not need MHC presentation. They can directly recognize their ligand and being activated directly. They have a faster response. They also possess strong cytotoxicity, so they can kill. And in addition, they can present antigens through MHC class 1 molecules to further activate other alpha beta T cells. So they also have regulatory functions. So in terms of killing, okay, so they can either kill through cytotoxic granules like CD8 T cells and NK cells. They can also kill through phosphates ligand and trail pathways. They can also do ADCC like NK cells. As I mentioned, they can present antigen. They can also produce cytokines to regulate immune response. So far, here are the clinical trials that been targeting gamma delta T cells. And you can see that overall it's quite safe. Okay, so the safety profile has been very good, but the objective response rate is only around 15%, and almost there's no complete response. So it's a bit disappointing at the moment, looking at gamma delta T cell adoptive therapy in cancer. You know, the one problem is similar to other adoptive therapy is the in vivo proliferation survival of those cells. Although you can expand them, activate them in vitro, once they're inside the patient's body, and they may not survive as long as you wish. Again, they also face immune suppressive tumor microenvironment. And it's also a concerning issue that people have found there are immune suppressive gamma delta T cell subset. So, you know, similar to T cells, right? We have regulatory T cells. I think in the gamma delta T cells, there are also subsets. For example, the IL-17 producing gamma delta T cells. We also published a paper a few years ago on that, that to show that those are actually immune suppressive because they can recruit myeloid-derived suppressor cells into the tumor microenvironment and then promote their function by producing IL-17 and then suppress T cell function. So this is also a concerning, you know, in terms of the cellular product you're making, you don't know what you have there, right? So I'm going to show you an effort in my lab done by a postdoc, Song Yuan, and a PhD student, and also my lab officer, Hui Yi, trying to make a better gamma delta T cell product by trying different combinations of cytokines. So the effect of different cytokines on gamma delta T cells are not very well studied. There are a lot of studies on alpha beta T cells, but still people have done quite minimal studies on combinations. As I mentioned, NK cells is probably the one that people have done the most studies with the cytokine combinations. So we want to ask, can the cytokine or cytokine combinations promote the anti-tumor function of the gamma delta T cells? So what are the best combination that we could use to promote their function, proliferation, survival? So they did a quite comprehensive screening using single, double, triple, and quadruplicates of IL-12, 15, 18, and 21. Those are the cytokines that have been shown singly to promote gamma delta T cell function. So what we found was that the IL-18 can enhance CD69, which is the early activation marker, and IL-12, 18 synergistically increase CD25 and interferon gamma production. So then we decide to keep the 12-18 combination and then include 15, 21 singly or all four together. So in the following experiment. So you can see here, this is a 48-hour proliferation assay. All four combinations promoted gamma delta T cell proliferation quite significantly. And in terms of you know, molecule expression related to killing, so perforin is also significantly increased, granzyme B slightly increased, and in TNF alpha slightly increased, and most importantly, it actually decreased IL-17, which is an inhibitory cytokine, at least, you know, in our tumor models. So the most striking outcome is the interferon gamma production. So it increased from around 20% to almost 100% of gamma delta T cells producing high level of interferon gamma. So this is percentage, this is the expression level, the MFI. So also increased dramatically. So then we think, you know, can they kill better? So we look at the killing assay here, short-term 4 hours, look at killing of a leukemia cell line and the liver cancer cell line, especially for the solid tumor. Actually, they kill a lot better with all four combinations. And the degranulation marker CD17A also increased for all combinations. So this is the 48-hour killing assay. So you can see all 4 combinations increased their killing capacity over a long time period. So this is quite promising. Then we decided to confirm that in the tumor in a human setting. So for human gamma delta T cells, and the one I've shown you are from the mouse gamma delta T cells, okay? Because we want to confirm that, so we could do the in vivo studies with the mouse model. So again, for human gamma delta T cells, that mainly, as I said, the V delta 1, the V delta 2. The delta 2 is the one mainly present in the blood. So we use 2 expansion strategies. So the antibody-based expansion can give you both delta 1, delta 2. But if you stimulate with phosphoantigens, the adrenic acid, and you get majority V delta 2. So we want to ask, do human gamma delta T cell respond similarly? And also these 2 human subsets, the delta 1, delta 2, and also different expansion strategies. Even we expand V delta 2 with antibody or with phosphoantigen, do they behave the same? So we look at the surface marker expression. CD25 increased, NKG2D, which is the activating receptor, also increased a bit. TNF a little bit. I mean, similarly to mouse gamma delta T cells, the interferon gamma production increased from less than 10% to almost 100%. So with antibody expansion, you get both subsets. With the phosphoantigen expansion, you get almost all V delta 2 cells. So we compare the different subsets. So CD16 is not much of a difference. TNF alpha slightly increased. Again, interferon gamma dramatically increased for all, for both expansion, for both subsets. But you can clearly see that the phosphoantigen expanded V delta 2 cells had the highest interferon gamma expression or production of all 3 cell populations that we examined. So for killing, all combinations increased killing capacity compared to the control. But you compare, this is the KPAT62, this is a liver cancer cell line. And although they all increased significantly, but you can tell the zoledronic acid expanded population had the higher killing capability compared to the antibody expanded cells. Okay. All right, then we try to test it in vivo using our liver cancer mouse model, which is orthotopic model where we have tumor growth in the liver of the mice. So we adoptively transferred all 4 populations at 1 week and 2 weeks. So compared to control gamma delta T cells, the cytokine pre-activated T cells, only the IL-12, 18, 21 significantly reduced tumor growth. So although there's 1 star, you can say this is not very impressive. Okay, although we did 2 adoptive transfer here. But we still decided to go ahead and look at the proliferation and apoptosis of the transferred cells. We use a label, fluorescent label, a CSFE. So when the cell proliferate and they lose the labeling, okay? So you look at the labeling here, the cytokine activated gamma delta T cells are losing their expression, the labeling here suggesting they're proliferating a lot better compared to the control cells, okay? So this is the quantitative analysis. Then we worry about their apoptosis. So although, they're super active, they're proliferating, producing a lot of interferon gamma, they also dying. In the spleen, overall it's less than 2%. Early apoptosis is very minimal. In the liver, there's actually a slight reduction here with the cytokine stimulation. So it's not really that of an issue here. Not only we have more, right? So we have more cells and they proliferate better, and they also produce more cytokines. So interferon gamma definitely, maintain their production, higher production in vivo, both in the spleen and in the liver. So not only, as we mentioned, that they also have regulatory functions, right? So if you look at the host CD8 T cells in the liver, and they also increase their interferon gamma production when we have the triple stimulated gamma delta T cells adoptively transferred. So there is an enhancement of the host cytotoxic T cells there. So in terms of mechanisms, right, the gamma delta T cells can either activate CD8 T cells by producing cytokines, or they could present antigens, right, through MHC and peptide presentation. So we decided to look at whether this is a cytokine-mediated event or you require cell-cell contact. So we put the CD8 T cells at the bottom of the trans well, and then we put gamma delta T cells on the top, on the upper chamber with the tumor antigens. So if they need antigen presentation, and they got the antigen there. So after co-culture here, you can see the one with contact, there's increased CD8 T cell proliferation. With Ki67 staining, there's increased early activation marker expression, and then the CD64 expression also higher. So if you look at the one with no contact, there's very minimal increase here, suggesting you do need cell-cell contact. So similarly with interferon gamma and TNF production, also in the contact group, the activation is more dramatic compared to the no contact group. So this is a activation of host CD8 T cells through a cell-cell contact dependent manner. So we're still trying to figure out whether this is MHC class 1 dependent, which requires antigen presentation. Okay, so for checkpoint blockade therapy, so you probably all know that PD-1 expression on T cells can bind to PD-L1 on the tumor cells, leading to the exhaustion or energy of the cytotoxic T cells, which can be a problem in the tumor microenvironment. So by using this antibody to block this interaction, and you can have a better T cell activation. So this is the idea for checkpoint blockade therapy in cancer. But however, in a lot of solid tumors, this therapy has shown very low efficacy. Okay, so even the one that actually better, you still have lower than 40% response rate. So now the field is looking at combination therapy to trying to improve checkpoint blockade therapy and the outcome of the overall cancer immunotherapy. So we decided to, since our gamma delta T cell single therapy is not that impressive, so we're also a bit worried that because this gamma delta T cells also have PD-1 expression, okay, so they could be easily inhibited by this PD-1, PD-L1 interaction too. So we decided to combine that with the PD-L1 antibody therapy in our mouse model. So here you can see with single checkpoint blockade therapy at this dosage that we chose, 100 microgram, there was no effect in our model. If you do the gamma delta T cell transfer alone, there's reduction of tumor growth, but still, as I mentioned, not very impressive. But if you combine the two, the red bar here, you get a synergistic effect of the two therapies, have a significant reduction, very good result actually in the outcome of tumor therapy. So, we look at the host CD8 T cells. It's surprising that only this group, right, had an increased interferon gamma production in the CD8 T cells that actually correlates with the outcome of the therapy. So just a brief summary on the the result I've just shown you, that the cytokine pre-activated murine or human gamma delta T cells have increased their activation marker expression, cytokine production, cytotoxicity. And then we found this phosphoantigen stimulation is probably better than antibody. V delta 2 is better than V delta 1. And then in our animal model, this combination therapy really enhanced the CD8 T cell function and had a better outcome. So we're thinking this, strategy of using this cytokine combination to enhance gamma delta T cell function is probably a promising way to go to boost gamma delta T cell function and overcome the resistance of immune checkpoint blockade. So just a quick note about the subsets. As I mentioned that there could be also different subsets of gamma delta T cells. We are still kind of working in the blend. We don't know actually the product we are using, the detailed, the markers that we use are quite limited to distinguish different subsets. So using the single cell RNA sequencing tool now, we can have a better picture of those gamma delta T cells too. So we did this, and then we actually found a cluster of cells have a very high cytotoxic molecule expressions, including granzyme B. Out of all the clusters, you see this one is bigger, and this one is got a reddish color. So we now have markers to isolate this population. We actually compare the killing capacity. You see this red population had, a significantly higher killing activity compared to the rest of the gamma delta T cells. So I think to better understand, what they have, what kind of gamma delta T cells they are, would help us to design and develop strategies to culture, actually expand and generate better gamma delta T cell populations for cancer immunotherapy. So I'm gonna thank the people who did the work. Mei Yu and Moose did the NK work. Hui Yi, Song Yuan, and Yong Hao worked on the gamma delta T cells with the help of the rest of the lab. Nick's lab helped us with some of the mouse models, and Veronique's lab with the parabiosis and the lipid study. Also our flow cytometry call with Paul, and Jing Miao helped with the single cell RNA sequencing study. And also my collaborator in Jinan from Jinan University, also my funding support. So I'm going to take another 5 minutes, just very quickly, to tell you exciting news of a professional certificate in application of immune effector cells in cell therapy, designed and developed in collaboration between ACTRIS and NUS. So this new professional certificate program is going to be rolling out starting in Q2 this year, very soon. So we're tidying up the ends and trying to recruit very soon. So this PC will equip our learners with the following competencies. Will help you understand the process of immune effector cells with emphasis on CAR-T. And key steps involved. You're going to gain a better understanding of the regulation of immune effector cells and implications in the practice of cell therapy. Also to help you understand the quality control, manufacturing, also the application to patients, and also acquire some knowledge in the manufacturing of the cells, and also the considerations in the clinical cell therapy clinical research. So it's very comprehensive. It's really an effort trying to combine all the expertise and to present to you a very good quality program. So we have excellent speakers. Going to have a 13-week program. Both online, live, and asynchronous lessons. And there will be pre-recorded lectures, there will be live lectures, there will be interactive sessions. So mainly in the evening and Saturday morning to accommodate working professionals. So we hope to target to biomedical graduates who plan maybe to switch their roles in regulation, business, quality management related to cell therapies, because we feel there's a lack of workforce in this area. So a lot of startup companies in cell therapy area actually are looking for talents in this specific knowledge domain. So the healthcare professionals, doctors, nurses, maybe you're keen to learn more about cell therapy and its application. Okay, so we only require a bachelor's degree, and other things we're going to look at it on a case-by-case basis. So if you have further questions, please contact our CET people at NUS. You're going to get more detailed information very soon. Okay, so I'm going to end my talk here. Sorry, I'm probably a bit overrun. So I'm going to look at the chat first, see what we have here. So I think some of the questions have already been asked. So yeah, so I think we can share the slides view. Okay, so nothing else in the chat. So the Q&A, okay, I'm going to the Q&A. So Selvin asked, would it be possible to produce NK cells in larger quantity? Yes, it has been done. As I mentioned, NK cell adoptive therapy have been used in transplantation settings for a while, and also like starting from Prof. Cabana's work at St. John Children's Research Hospital, they have also done a lot of NK adoptive therapy there, so it's feasible. So there are quite a few clinical trials. If you go to clinicaltrials.org, you can see, yeah, there are some in Singapore, mainly by Dario Cabana's group, done at NUH. So what are the challenges? I think I mentioned some of the challenges there. I think the biggest challenge is to overcome the immune suppressive tumor microenvironment, and also try to prolong their survival and maintain their effector function in vivo, right? So there's one from WeCat. There's a good increase in interferon gamma at the cytokine. However, would the cytokine cocktail also increase production IL-6 of gamma in vivo? Yeah, that's a good question. I don't think we have looked at IL-6 as, you know, on top of my mind, on top of my head, I can't remember if we look at it. I can't answer that. But it is a concern if IL-6 is high, you worry about CRS. So, but, you know, from our preclinical study, the mouse study, we don't see any toxicity. Actually, they seem very safe. After 2 transfer once a week, we don't see any toxicity in the mice that we treated. Also in the clinical trials, like when people try to stimulate gamma delta T cells, and as I mentioned, the safety profile has been very good. But, you know, again, with this cytokine activation, we don't know. In the combination therapy with Chelsea, did the gamma delta T cell persist? Yes, they do. So we can still see them actually at the end of the experiment, which is 3 weeks. They do increase interferon gamma production. Yes, I didn't have time to show you there. No, the PC is not free. Sorry, Ankit. And if you want to know about the cost, you can write an email there if you want to know in advance. But I think we're going to, publicly put out the information very soon. So William, you like to ask if it's correct to say NK therapy cannot kill MHC1 expressing cancer cells, and if so, have there been efforts to overcome? Yes. So this is this is a very legit question. For MHC class 1 expressing cancer cells, then there will be inhibitory signal there, right? So that's why in the I group them into the checkpoint therapy, where the anti-KIA or other antibodies can be used to block this MHC class 1 recognition. So then the NK inhibitory signal will not be transferred, so then NK cells can kill without the inhibition from the MHC class 1 expression. Yes, that can be done. How feasible to obtain? They are they can be very easily expanded from PBMC. Yeah, so you can get a huge number of gamma delta T cells from PBMC. And also, we're also trying to look at other sources. I know that you could try from cord blood. That's another source for gamma delta T cells. Can you explain briefly how PD-L1 antibody work for tumor therapies? Okay, I'm sorry, Arzila, maybe I went through it too fast. So the PD-1 expression on T cells can transduce inhibitory signal in the T cells, so it can inhibit T cell function. So a lot of tumors actually use this mechanism to suppress T cells. They express PD-L1, which is the ligand for PD-1. So PD-L1 expressed by tumors can bind to PD-1 expressed by T cells and then suppress T cell function to transduce the PD-1 signal. So if you have an antibody which is a blocking antibody that can bind to PD-1 or PD-L1, then you can basically block this interaction. So by blocking this interaction, and you don't have PD-1 signal in the T cells, so your T cells will not be suppressed. So this is the way to better activate T cells in immunotherapy. Okay, so from Andy, figuring out which subset of NK gamma delta T cells are ideally cytotoxic, long-lasting instead of immune suppressive. Using single cell RNA-seq is great, but how does one translate this to manufacturing these ideal subsets for clinical translation? That's a fantastic question. It's a long way to go, but first we have to understand what they are, right? So once you know this subset, right, either the one that's highly cytotoxic or the one that you see that being suppressed in the tumor microenvironment. So for the one that's highly cytotoxic, you can try to maybe look at the pathways, see which pathway is specifically activated in those cells. Maybe during the manufacturing process, you can add reagents to activate that pathway. So then you can generate that population in vitro. I'm not saying that it's 100% possible, but it's a direction to go. That's where we're aiming to. So once we understand fully of that T cells, gamma delta T cell NK subset, and we can manipulate their either their signaling pathways or using their specific surface markers to isolate them. Either you isolate them out or you manipulate to expand them in vitro. Yeah, I think that's the way to go. So if you see certain subsets that are specifically induced in the tumor microenvironment, and you can also design, for example, antibodies to block that process. So once the NK cells are in the tumor microenvironment, they don't get suppressed. They don't get induced into that immune suppressive subset. So this blocking mechanism can also be used to prevent, right, NK suppression or gamma delta T cell suppression. What is the negative control for gamma delta T cell killing assay, since they can kill all types of tumor cells? Yeah, they do. They have pretty good killing capacity. Yeah, you're right that we should look at more normal cell lines to see if this killing is tumor specific. We did try some, and I think they are they're not totally specific, but they do have a heightened killing capacity against tumor cells. As for the negative control, I think maybe that's what you mean. Yeah, but then we have the control with the control gamma delta T cells, which are not stimulated with the gamma delta with the cytokines as our negative controls. Do you foresee that gamma delta T cell from xenograft sources cross individual transplant to work well? So I guess you do not mean xenograft. It's you mean the allo the allo one, the allo react, from the allo cells, not the autologous, right? Yeah, so it is possible because they're not HLA restricted. But I think this is the field that we also need to be a bit careful, because I think NK cells have been shown to be able to survive in a different individual in a lot of allo NK studies that have been done. For allo gamma delta T cells, that there's very little information there. People haven't really done much of the allo gamma delta T cells. So I think we need to wait and see how how much is the rejection there, because they're probably more immunogenic than NK cells, and I'm not sure. But the allo NK seems to be well tolerated in the allo settings. Yes. Okay.
Thank you, Prof.
So I cleared all the questions.
Yes, Prof. Liu, thank you so much. So thank you, Prof. Liu. We have come to the end of our lecture. On behalf of ACTRIS, we would like to thank you for your time today, and we hope to see you again in our next lecture on 16th of February. As you sign off, you will be asked to answer a short survey pertaining to the webinar. We will appreciate any feedback you have for us, as it helps us to be better at what we do. Once again, thank you everyone, and have a good day.
Thank you. Okay, thank you. I'm going to leave now.
Thank you, Prof. Liu. Goodbye.
Bye.
Hi, Shani.